Hello class, Mr. Tanderson here. Today we'll be learning about transcription and processing in the first half of this video, and translation to follow shortly thereafter. Sure you're curious as to why I've included this fine pair of pants right here, but fear not, I'll be explaining in time, so let's get into it. Here we see a picture of a DNA strand, and our buddy here, RNA polymerase II, trying to attach to the DNA strand. Now it manages to pull this off with help from its friends, the Tata box, the transcription factors, and the promoters. This process occurring includes the initiation phase of transcription. From this point on, the RNA polymerase II, once it's all settled into its new home here, it pumps out the inklings of our messenger RNA, which is pre-mRNA. This process is known as elongation. Now it's important to note here the 5' 3' direction of the DNA strand, because that comes in important later. Next we move on to this slide. And here we see a new player on the field. We see the SNRNPs. Now remember our pre-mRNA strand from earlier? Now this guy binds with him, and it creates a spliceosome. Like a little guy in here. The spliceosome does is he cuts out the introns. Now these guys do not get expressed. The exons are the ones that get expressed. A good way to remember the introns are like shunned family members. You don't want anyone to know about them. You kind of keep them locked in the basement, out of sight, out of mind. Feed them every now and again. But now with the exons, you want them out there. You want everyone to know how awesome your family is, or at least for them to think your family is awesome. So you just show off them and their accomplishments, you know. Those are the exons. So then you got your introns cut out, your exons ready to be shipped out and ready to go. And that brings us here. Let's see if this slide will get moving. Come on, slide. You can do it. There we go. Alright. So then that brings us back to our pants. So remember that messenger RNA we had earlier? That's like this guy, right? And that's going to be where he goes. So that those pants are going to zip back up because you don't want your fly down in public. That would just be outright embarrassing. So then let's take a look at the structure of this mRNA. Here on the tip of the mRNA, we've got a myelated G cap at the 5' end. Now that's important because it promotes translation, which is the whole point of this process. And on the end we got the A tail, poly A tail. Now, see, it's important because once it's going off in the cytoplasm, it gets degraded by enzymes. Now, a good way to remember the A' tail is, say you're out in the woods, right? So you gotta make sure you bring along a line behind you. People you're willing to sacrifice, people you're not entirely that concerned about. Like you're kind of sort of friends with them, but they're not that important to you. That way when you get attacked by bears or wolves, you can just leave them behind and they can die without you really losing anyone too important. That's a good way to remember that. And then bam, you're ready to go. You got your mRNA all excited to go out and be a strong, independent thing in the cell and do its job, or one step closer to that anyway. For now, that will bring us to our that'll bring us to intermission. So let's get into that. I'm always excited for intermission. Smooth jazz going. Oh yeah. Just absorb that information, let it all in. You know, music proves, or research proves that music's good for learning. So just let that knowledge soak in, really internalize it. Transcription, translation, yeah. I guess we should get back into the actual learning bit of the video. It's always difficult to leave intermission. Alright, that brings us to translation. Here we go. Remember our little buddy, the mRNA from before. Well, this is him. Here he is. Tracing 101 with Mr. Tanderson. There you go. So you can see we have our buddy, the mRNA, sliding through the ribosome, which consists of two parts, a smaller subunit on the bottom and a larger subunit on top. Now, a very important component of the ribosome is the rRNA, which is an essential component of it, which lets it function really and allows us to create proteins, which is obviously very important. Something to note here is that 3 letters code for one amino acid, and our initiator tRNA, this guy right here, binding to our start codon, AUG, forms the initiator complex. When that brings us to our sites. We have 3 sites, the A site, which is for the acceptor site, P, which helps the tRNA bind to the mRNA, and then E, the exit strand. That's all some pretty fantastical stuff. So then that brings us to, this slide will change. Come on, slide, you can do it. I have faith. Come on. Come on, let's go. I need to switch to that slide. Come on. Aha, there we go. Alright, finally got that to switch. Alright, so here we can see on our sites we got our A site, our P site, and our E site. A little bit more of a detailed look. We see our AUG, our start codon here. That's all pretty fantastical stuff. So maybe a little simile or metaphor would help us figure out and better understand. And here we go. See, we got our explanation here, the codon recognition. Now, how does that work? It's like a bit like a puzzle. So let's say we're working with our start codon. Our A, U, G. Excuse the poor penmanship, even on the computer. Now, that's our codon, and our anticodon for that would be U, A, C. That's just how that'd be like an amino acid, you know, the very first in our chain. Now that brings us to, get here, come on, you can change, come on. Maybe get him to switch here. A little difficult, technical difficulties, excuse that. I just have to do it without the picture if it's not going to cooperate. There we go. Alright. So we got our tRNA synthetase here. Here we've got the AMP and the ATP, which binds the tRNA with our pyrophosphate group. Once that releases, the tRNA binds to the AMP and ATP and forms a bond and creates a high energy bond to tRNA. Then we got our peptide bonds, peptide bond formation, which attaches the amino acids to each other, which is very important for the construction of our polypeptide chains. And the fuel for all of this is GTP, which is the energy source for binding the amino acids together. Now, all good things must come to an end. Even this. So, we got our chain of amino acids built up, and then it only stops once it hits a stop codon. As Arnold Schwarzenegger would say, that would be terminated. And that would be termination stage completed. Once the stop codon's done, and then once it hits the release factor, we got our chain of amino acids and proteins just flying free, home free in the cell. Kind of reminds me of the snake charmer, you know? You got the amino acids going up there, and then they break 3 and slither around in the ocean. Or the poisonous snakes, you know? You got snake charmer, same general shape, and it pops off and bites you. It's ornery. Ah, that was a bit of a tangent, you know, that concludes the video. I hope that was helpful.